The process by which the body transitions from a state of heightened alertness and readiness for combat is a topic of interest. Greetings everyone. In today's video, we will be delving into the subject of glucocorticoids, such as cortisone, which are highly effective medications used to combat immune-mediated inflammatory diseases. An enlightening study conducted by Professor Gerhard Kronke has shed light on the fact that glucocorticoids not only impact gene expression, but also have a significant influence on cell metabolism, particularly within mitochondria. These medications have the ability to reverse the redirection of energy in immune cells, which is crucial for their anti-inflammatory properties. Additionally, the study highlights the importance of itaconate, a natural substance known for its anti-inflammatory properties and its potential in the development of new synthetic compounds that could offer fewer side effects. Without further delay, let us begin our discussion. Cortisone, a glucocorticoid, is a natural hormone called cortisol that exists within the body. When faced with stress, the body releases cortisol to enhance its response. Cortisol plays a role in regulating sugar and fat metabolism, as well as impacting various factors such as blood pressure, heart rate, and respiration. At higher doses, cortisol also hampers immune system activity, making it valuable for medical purposes. To effectively combat inflammation, synthetic glucocorticoid derivatives are utilized, surpassing the body's own natural cortisol in effectiveness. These medications are widely employed in treating a broad range of immune-mediated inflammatory conditions and are among the most commonly used drugs. However, it is important to note that glucocorticoid-based medications can produce side effects, particularly when administered at higher doses or over extended periods. These side effects are connected to the additional effects of the body's own cortisol. High blood pressure, osteoporosis, diabetes, and weight gain are among the conditions associated with the use of glucocorticoids. To address this issue, Professor Gerhard Kronke and his team at Charité have undertaken a comprehensive examination of the immunosuppressive properties of glucocorticoids. Their objective is to develop alternative anti-inflammatory treatments that have milder and less detrimental side effects. According to Kronke, it was previously understood that glucocorticoids have the ability to activate various genes in different cells throughout the body. However, this activation primarily targets the existing resources within the body, which does not adequately explain the powerful immunosuppressive effects of glucocorticoids. In our study, we have made a significant discovery by demonstrating that glucocorticoids have a broader impact beyond gene expression in immune cells. They also affect the mitochondria, which are the powerhouses of the cell. This influence on cell metabolism is crucial for the anti-inflammatory effects produced by glucocorticoids. To conduct our research, we specifically focused on macrophages, a type of immune cell responsible for eliminating intruders like viruses and bacteria. Additionally, it is worth noting that these cells can also contribute to the development of immune-mediated inflammatory diseases. In a laboratory setting, the researchers examined the response of immune cells, obtained from mice, to inflammatory stimuli and the impact of administering a glucocorticoid. Alongside its influence on gene expression, the researchers noted that glucocorticoids played a significant role in reversing alterations in cell metabolism induced by the inflammatory stimuli. According to Kronke, when macrophages enter a state of aggression, they redirect their cellular energy towards preparing for battle. Instead of providing energy, their mitochondria start producing the necessary components to combat intruders. This process can be reversed by glucocorticoids, which deactivate the aggressive state and transform weapons into tools for peaceful purposes. Itaconate, a small molecule produced naturally in the body's mitochondria, plays a crucial role in this process. When macrophages are activated, they generate itaconate to regulate the inflammatory response and ensure that it subsides after a certain period. However, the production of this natural immune brake requires an adequate fuel supply. When the cell's powerhouses are focused on preparing for battle, this supply diminishes, causing itaconate production to eventually cease. This timing is effective during normal short-term inflammation, because the immune response also subsides over time. Dr. Jean-Philippe Auger, a scientist at the Department of Medicine, Rheumatology and Immunology at Uniklinikum Erlangen, and the first author of the study, explains that when there is a persistent inflammatory stimulus, the decrease in itaconate production becomes problematic. This is because the immune system continues to operate at full capacity without any break, leading to the development of chronic inflammation. However, glucocorticoids come into play in this situation. By reprogramming mitochondrial function, they enhance the production of itaconate in macrophages, thereby restoring its anti-inflammatory properties. The researchers conducted experiments using animal models of asthma and rheumatoid arthritis, which clearly demonstrated the significant role of itaconate in the anti-inflammatory effects of glucocorticoids. Animals that were unable to produce itaconate did not respond to glucocorticoid treatment. These findings raise an intriguing question. Instead of administering glucocorticoids, could we potentially directly administer itaconate to achieve its immunosuppressant effects? Regrettably, itaconate does not possess the necessary qualities to be considered a viable option for an anti-inflammatory medication. Its instability and high reactivity make it unsuitable for systemic administration, as it may result in adverse reactions. Additionally, the complexity of human processes compared to those of mice necessitates the search for alternative synthetic compounds. The goal is to identify new substances that can effectively reprogram the mitochondrial metabolism within immune cells, similar to glucocorticoids, while minimizing the occurrence and severity of side effects. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.